Okay, so hello and welcome everyone to the live session of week 7 on the course Experimental Biochemistry taught by Professor Shagota Dasgupta and Professor Shoumodaya of IIT Kharagpur. I am Srijan Sen and today I will be taking your live session on the lectures which were taught to you and provided to you for the week 7, and also solving a few assignments and questions which would be helping you in solving your assignments and the upcoming exam you people would be having. Okay, so I'm sharing my screen. I guess my screen is visible to you people. Okay, that's good. We can start then. So in the last class or in the classes before, we discussed about a lot of about proteins, amino acids, and the ways we can purify proteins. So today we would be mostly discussing about the isolation and the characterization of the proteins, as to how we can isolate the proteins, what are the different means by which we can isolate proteins, and then we would be discussing most about what we discussed in the previous classes as well, about how we can characterize proteins and purify proteins, and in what manner we can observe proteins. So I will be starting with the isolation of proteins. So how can proteins be isolated? So basically proteins can be isolated from two main sources. One can be from your natural means, that can be from your either be from plants or some mammals, or provided by some prokaryotes or eukaryotic organisms. And the other way of isolating proteins is by the recombinant means, that is there are different recombinant proteins, and we actually change the nature of the proteins to isolate them. So these are via the different recombinant sources. So the first one is via your natural sources, and the next one via your recombinant means that can be expressed from your different sources. So basically, how can proteins be isolated from natural sources? It includes your different, say your different tissues, then your plants, then your embryos, then you have some culture tissue cells, or it can be taken from the whole organisms or microorganisms. These are the natural sources. Or some organisms naturally produce some proteins from which these proteins can be isolated naturally. The other way of saying is that proteins can be isolated by recombinant means, that is, which are expressed from the different sources. So these includes your fungi, then some kinds of plants, then we have the mammalian cells, or some animals which produce different whole animal production systems, sometimes insects and microorganisms. So these are the various ways of isolating the different proteins. Then this is the, you can observe, is a cell, and it has different parts and the different organelles present in it. So we are not concerned about it at present. So what we do is mostly important. So the thing is that what is the general strategy behind our protein purification? So first we have to take a microorganism, or say a plant, be it from the naturally occurring or via the recombinant means, and then what we do, we extract its say its tissue, or the entire microorganism, and then what we do is disrupt it, or break it apart. So here we can see that we are breaking it apart and actually extracting the crude from it, or the materials from it. And after disrupting the cell, we get the entire organelles into our solution, and via which we can undergo some crude fractionation, from which we remove the unrequired or the unnecessary parts, and then via our selective fractionation we can extract our main protein. So this is the general strategy. So this disruption is actually telling us about the disruption of the outer membrane, or we sometimes we call it the cell wall. So we are breaking apart the cell wall, bringing out all the organelles including our, say, nucleus, then we have the cytoplasmic membrane, then we have the ribosomes and other you have other organelles present inside the cytoplasm. So these comes out into our solution, and then we undergo some kind of centrifugation or fractionation, and then say after giving a hard spin or a hard centrifugation, that is a centrifugation at higher rpm values or a higher at a higher speed, then we actually remove some of these organelles, and then via our, because some organelles will be actually separating out at very less range of your centrifugation value. So it is important to actually undergo also selective fractionation for the removing or actually you say separating out these close apart organelles, and via this means we get our actual protein of interest. So first we do is isolation, then disruption of the cell, and then via the different means we get our protein of interest. Now, the different protein isolation methods includes, first of all, is when we isolate our proteins which are actually varying on solubility. So we perform out the salting out experiment, where we actually remove the salts from our proteins, because some proteins are soluble in different salt conditions, or okay, so some proteins are soluble in different salts, and when you actually take out the salt, the protein precipitates. Or when you actually give it some more salt, what happens? The protein actually is not very comfortable in such a solution. So one of the process of protein isolation includes your salting out process. The other process is your centrifugation, where in when we actually want to get our proteins in vitro. Okay, so we give growth of our say the glycerol stalk. From the glycerol stalk or from the colonies we pick up a colony, and then we give it a growth, and then we grow it on the lurea botany broth, and after which what we do is we lyse the cells, then do sonication, and after that we give it a centrifugation, where we get the pellets out and the protein remains in the solution. So this is another way of how we can isolate our proteins. I will be speaking about these topics in details, but as of now you understand centrifugation is actually via the process of centrifugation we are removing the unnecessary parts and getting the proteins after the process of lysis, or after we have lysed the cells, disrupted the cell walls, and the entire thing of the from the bacteria or the eukaryotic cell is out, and from the cells are out, and then from that we actually separate our proteins. So this process which I was talking about, like in the bacteria, from the bacteria when we get our proteins, there are different conditions, like we give IPTG such that the bacteria is able to produce our protein of interest. Okay, so there are different ways. So the second process is centrifugation. The third one is chromatography. So what we do, so there are different ways in how we do. So as I was speaking about this in the previous classes, as well as how we can separate our proteins on the basis of size, then mass. So we do this using what type of chromatography? It is the molecular sieve chromatography, or we call as the gel filtration chromatography, or the gel permeation chromatography. Then if we want to separate our proteins on the basis of, say, charge, then we perform is ion exchange chromatography. Or if there are too many number of hydrophobic residues present in our protein, and we want to separate it out from a protein having say a greater number of hydrophilic regions. So how do we separate it? We separate them on the basis of the term as hydrophobicity, that is via the hydrophobic interaction chromatography or the reverse phase chromatography. And if we want to separate them on the basis of the binding affinities, then the name itself suggests that we perform affinity chromatography or antibody chromatography, where there is binding of the antibody to your particular proteins, and then we detect it under some kind of fluorescence, or say we give it a substrate, and then that substrate is oxidized or reduced, and then we get a color. Then we can observe it visually, or sometimes we can observe it under some kind of fluorescence channel. So there are two types of by which we can separate our proteins on the basis of binding affinity. One is our affinity chromatography, and the other one is our antibodies, by the use of antibodies. So another process by which this isolation of proteins can be done is via a selective precipitation. So this selective precipitation depends on some of the major factors, which includes our ionic strength. Then we have some pH conditions. Then some kinds of, by changing the ionic strength or the changing the pH, then some kind of selective precipitation occurs, and then we precipitate out our protein, and then provided we can isolate our proteins in different conditions. Also we can do it via using different organic solvents, and also by using some molecular crowding agents. So molecular crowding agents is we are crowding our proteins with different kinds of chemicals or some kinds of agents which we use, one of which includes our polyethylene glycol. So this can be used by, when the protein gets overcrowded, then what happens, it gets isolated out, or it precipitates. So we can do this by different kinds of agents. So the first one is our ionic strength, then the pH. So the pH, how the pH plays a role. So we do this when so we do this by on the basis of the pI which the protein has. Okay, so these things can be done. And then we have the organic solvents, and then some molecular crowding agents, which is one of which is the polyethylene glycol. So what it does, so what is the selective precipitation, or in what manner, or what type of proteins can undergo this selective precipitation? One which has the specific property of, say, our target protein. And then when the proteins vary on the basis of our charge and hydrophobicity, and if the proteins have different solubility and size. So in this manner, actually we can selectively precipitate our proteins and isolate our proteins from the other proteins or the protein of our interest. The next process which we can take over is via our salt fractionation. So what is this process? So here comes out the concept of our salting in and salting out. So what happens over here? So we see that when there is a mild increase in the salt concentration, say a protein is in a particular salt concentration and it is soluble over there. You're not concerned. So this is our protein of interest, say, so this one. Okay. And so this one is our protein of interest. It is present over here. Now, a very minute or very mild increase in the salt concentration actually causes a low ionic strength. That is, when we are increasing the salt concentration, it is causing low ionic strength, and this is leading to increase in the solubility. Okay, so the protein becomes more and more soluble even. And what happens is that when solubility increases because of the mild increase in the salt concentrations, then the salting in process occurs. Whereas on the other hand, at a very high salt concentration, when the salt concentration is very, very high, then there is a decrease in the solubility as the ionic strength increases vigorously. Okay, so when the ionic strength increases vigorously, we can see over here that there is a crowding of the ions around our protein of interest, and then the salting out phenomenon occurs. In both of these cases, what happens is that the protein can precipitate out. Okay, or this is the process by which salting in and salting out occurs. So when the protein is in this condition, it is very you can say it is soluble enough, but a minute increase causes the salting in, whereas a high salt concentration increase will actually cause the decrease in the solubility. When when there is an increase in minute amount of salt concentration, the protein is soluble enough, and the solubility actually increases, resulting in the salting in phenomenon. Whereas when there is very high salt concentration, there is the decrease in the solubility, and because of the increase in the ionic strength, and thereby the salting out phenomenon occurs. So there are some ions which are more effective than the other ions in your protein in affecting the protein solubility. So there was a scientist named Hofmeister who gave these series as to how the salting in or the salting out, or which ions could be included in these two phenomena. So these series can be termed as your Hofmeister series, because some ions will actually be very good in the salting out phenomenon, such as your ammonium, then your potassium, or mostly your positively charged ions would actually be stabilizing. So these have a stabilizing condition, and actually stabilizes your proteins and helps in the salting out phenomenon. So ammonium is greater than your potassium, then your sodium, lithium, magnesium, calcium, and finally your guanidinium. And for the salting in phenomenon, that is, these have a denaturing property or the salting in phenomenon. So then we have your sulfate, which is greater than your phosphates, then your acetates, chlorides, iodides, and your thiocyanates. Okay, so depending on these, the combination of these two can be actually used for whether you want to salt, can undergo your salting out or the salting in phenomenon altogether. It's like we have when we see in this graph, we can see that this is ammonium sulfate, then we have potassium fluoride, then we have guanidinium hydrosulfate, then potassium chloride, then ammonium chloride, and the guanidinium hydrochloride. Okay, so these can be actually, from these graph we can tell which undergoes better salting in, or which can be used better for increasing the solubility or precipitating out proteins. So what is the mode of action of these salt fractionation method, or what do we do, or how this actually works? So we see that high concentration of small highly charged ions. The higher concentration of a small or highly charged ion, such as the ammonium sulfate, it competes with the proteins to interact with the water molecules. So when there is higher concentration of small charged ions, it actually, what it does, it actually wants to fight with our proteins, and then the competition is on as to who wants to interact in what manner with the water molecules. Now what happens, when this competition is actually going on, the water gets removed from the proteins, because after all the increasing concentration would actually be good enough, the competition would be good enough for these salts or these ions to remove the water molecules from the protein. Then what happens, when the water molecules around the proteins decreases, the solubility actually decreases, resulting in the precipitation of the proteins. So if we say in the other way, so increasing concentration of your small or highly charged ions would actually remove the water molecules from the surface of the proteins or from the environment of the proteins, leading to the decrease in the solubility. So increasing concentration of small highly charged ions decreases your solubility and increases your precipitation. Okay, so these is important, that high concentration and then comes your decreased solubility and increased precipitation. So now coming to the next technique, which is a centrifugation. So we have seen that centrifugation works via different, and there are different kinds of centrifuges which we use. We have the swinging arm rotor, or sometimes we have the tabletop centrifuge. Then we have sometimes the fixed angle rotor. So the first one is the fixed angle rotor, where the angle between the two where we put the tubes or the things uh, is actually fixed, and it has a fixed angle in between them. Okay, so this is a fixed angle where the angle does not change. Not change much is wrong. Yes, it does not change at all. Okay, so then these type of centrifuges can be refrigerated ones or your non-refrigerated ones. So the refrigerated ones are if you want to get your protein in 4 degrees or at a degrees temperature or at an increased temperature, anything as such, or at a particular temperature, then these things can be refrigerated, not at room temperature. Okay, so there so these are the different types, and then when the angle between the two stands are fixed, it is called the fixed angle rotor. And then the other type is the swinging arm rotor, that is when the arms of these actually swing around. So how they do? So when the centrifugation is on, or when you switch on the centrifuge and start it, these rotors actually have the ability to move and form an angle. So this has an angle of 180 degrees. Okay, so these are very parallel to each other and then moves. So mostly cell fractionations are done in these type of rotors, because these hold the tubes. These are some metal buckets which hold the tubes and then allows it to freely swing outwards as the rotor turns. So this is not a free this is not a fixed one, and hence it has the ability to turn around and form an angle. Okay, so the centrifugal force is very well faced by your at the end of the tube, and you would be getting good amount of what you say, the unnecessary or the pellets at the end of your tube, and you can actually take the protein from the supernet. So this actually gives you good so we can call this a good centrifugation technique, and you get good amount of proteins from there. But what happens in the fixed angle rotor is that you get your pellets along the sides, and sometimes if you can see from this picture, this is your pellet, but you also have some kind of collections out above your pellet or along the sides of your proteins that is present in the supernet. So it is very difficult sometimes to remove the proteins from the supernet. Whereas on the other hand, if you can see that you get only a very concentrated amount of pellet along the bottom of the tubes, which would also help you in better and getting pure proteins, better and pure proteins also. Okay, so what you do before the centrifugation? You have a cell homogenate where you have the proteins and then the other kinds of cell debris, then your pellets, unnecessary proteins as well. Okay, and then when you undergo centrifugation, you get the supernet, which contains mostly your smaller and less dense components, and the pellet contains your larger or the more dense components after you undergo centrifugation. So if there is a question regarding the centrifugation as to which centrifuge should one person be using, so it is not that people do not use the rigid or the fixed angle rotor centrifuge. There are different things, or different ways in which you want to separate, depending on which we use the different fuges. Okay, or the different fuge. And so there are different ways, but the best one to use it is the second type, or the swinging arm rotor, because it gives you pure supernatant, and the pellet is settled mostly at the bottom of the tube, which will help you in taking out your supernatant from the top and performing a reaction or whatever you want to do. So this is your cell homogenate, and you undergo, say, a low speed centrifugation. So mostly what would happen, the what separates out. So firstly, okay, so the cell homogenate mostly contains, so first what you have done, you have taken a cell, say a normal cell, and then you disrupt it, and then you get the cell homogenates, and after which you give it a very low spin and a low speed centrifugation. So what happens in the pellet, what you get are mostly your whole cells, then you get your nuclei and the cytoskeletons. Okay, so these three you would be getting after you give it a low speed centrifugation, because your pellet contains mostly your larger and the more dense components. So these are the larger components, and you would be getting these even at low speed centrifugation. Now what you do, you take out the supernatant, you take the supernatant out and give it a medium speed centrifugation. So the medium speed centrifugation would actually separate out your, means your pellet would contain mostly your mitochondria, your lysozymes and your peroxisomes. So these are the different organelles which under medium speed centrifugation will come to your pellet. And then what you do, you again take the supernatant from the top, and then you give it a very high speed centrifugation, and then what you get is mostly your microsomes and the small vesicles, which are less dense, which were less dense in the beginning. So it needs to give in some kind of high speed centrifugation, and then you get your microsomes and the small vesicles. Again, you take your supernatant from the top and give it a very high speed centrifugation, and then in the pellet you get your ribosomes, then mostly your viruses and large macromolecules if they are present. Okay, so this is important because these now I'm telling you it's low speed centrifugation, but these have some particular values. So these values actually matter, because there might be a question, uh, I don't know, but there can be a few questions like, or a question like, the cell homogenate is subjected to a centrifugation, say a medium speed centrifugation. As I've not spoken of the values, so let's say is subjected to a medium speed centrifugation. Then what would you expect to get in the pellets, or what organelles do you expect to get in the pellets? So this can be an MSQ type, and then there can be a few options given to you, like your whole cell, like your mitochondria, lysozymes, peroxisomes. Also, when you undergo medium speed centrifugation without the low speed centrifugation, then you have to also consider the whole cells, nuclei and cytoskeletons as well, because in the first step only you are undergoing the medium speed centrifugation without even separating out the nuclei and the cytoskeleton. So you have to read the question very clearly and see what the question is asking from you. So once more I'm repeating, when you undergo a low speed centrifugation on the cell homogenate, you get your whole cells, nuclei and cytoskeletons in the pellet, and then you take the supernatant from the top and then undergo the centrifugation, but at a medium speed, and then you get your mitochondria, lysozymes and the peroxisomes in the pellet, and after which you give it a high speed centrifugation, and then your pellet contains your microsomes and the small vesicles, and after a high speed centrifugation is done, then you get it, a very high centrifugation is done, then you get your ribosomes, viruses and the large macromolecules. Okay, now coming to the thing. Now what are the different conditions? It is like this. Now your homogeneity is present, and then you centrifuge it at say 500 G. So what are the different units of the speed? Can any one of you tell me what are the different ways we actually determine speed when we do it in a centrifuge? Any one of you? What are the different ways in which we can actually measure these speeds? So have anyone of you used any kind of centrifuges before? Okay, so if you have used it, you know the units probably, you can just tell, because then you have to set the speed. Probably if it is a refrigerated one, then you have to set the temperature. So different companies have different ways of actually separating, setting the temperature and the time you have to set. After that, you start the instrument. So how do you involve? What unit do you set the speed? There is 4,500 or 4,000 or 15,000, something like this. What are the units? No? Okay, so mostly the speed is set either in RPM, or you can use RCF or G. So these are the mostly or the commonly uh, used ones. Okay, so these are mostly used. So we can use RPM or G. So here we have done it in terms of G. So say the cell homogeneity is given a spin at 1000 G for 10 minutes. And then what we do is we collect the supernet, because your pellet would be containing your whole cells, then the nuclear fractions. So your low spin is actually your 500 to 1000 G for 10 minutes. So this is important, because then you have to know what is the speed. So this thing is important, like it can vary between 500 to 1000. So this is a low spin, and for 10 minutes you do, and then you get your whole cells, then your nucleus, and then what was that, your cytoskeletons. And then the supernet is the pellet. You keep the pellet or you discard the pellet, that's all upon you. And then you collect the supernet and give it a speed, a medium speed, and that speed varies between your 10,000 to your 20,000 for say 20 minutes you give it. So this is 10,000 to 20,000 G. And then what you get in the pellet is your mitochondria, then lysozymes and the peroxisomes, or you can tell the mitochondrial fractions. Okay, and then after this you again collect the supernet and then give it a speed, give it a spin at say 80,000 to 1 lakh G for 1 hour. Okay, and then what you get is your microsomal fractions, or you get your say microsomes and the small vesicles. And if you go above that, if your centrifuge can actually go above that, say 2 lakhs G or 2 million G for say 1 hour or 1.5 hours or 2 hours, say, then you would be getting your ribosomes, then the viruses, or the large molecules. Okay, so these values actually matter, as maybe you would be asked to say that your supernet, the cell homogeneity is subjected to say 10,000 G for 20 minutes. Then what would you expect in the pellet? Or say you are given a speed of 1 lakh G for 1 hour. Then what would you expect in the pellet? Say mitochondrial fraction, then the nuclear fraction. So depending on that, maybe in details you won't be asked as to say the whole cells, nuclei, cytoskeletons, or the mitochondrial lysosomes, peroxisomes, not much in detail, but maybe you can be asked to just tell whether you are getting a nuclear fraction or a mitochondrial fraction or a microsomal fraction. So any of this. So these values you can just keep in mind, would be beneficial. There might be, you expect a few questions, nothing else that. Okay. So the other different techniques which you can use is the ultracentrifugation technique, which is basically used for your finer separations or and your less convective mixing. We do ultracentrifugation techniques depend on the varying density. That is when we create a density gradient using our sucrose and some uh, heavy say heavy metals, say like cesium chloride, then or large atoms, then we actually create a gradient of density. Based on this, the finer separation or the less convecting mixing separations can be done using our ultracentrifugation technique. So the most important thing over here is, so ultracentrifugation technique actually takes the path of density gradients. It creates density gradients using sucrose or uses cesium chloride, and then we undergo finer separations. So the other technique which we can use is the sedimentation techniques, and this so sedimentation techniques can be two types, or in two ways it can be done. One is the velocity sedimentation, another one is the equilibrium sedimentation. So sedimentation undergoes, so it takes over the principle of your centrifugal force, where we have omega square Rm, where your omega is the angular velocity measured in radians per second, your R is the distance from the axis, and then we have M is the molecular mass. Also, in sedimentation, we have the different, so these are the different forces which actually play a role in the process of sedimentation. So the first one is your I mentioned is the centrifugal force. The next one is your buoyancy force, where you have minus omega square r m naught. So where your omega, I mentioned it's an angular velocity. Then you have r, which is the distance from the axis, and your m naught here is the displaced mass, so the amount of mass which is being displaced. So that will actually is equivalent to your buoyancy force. Then we have friction, which is your minus Fv, where F is your frictional coefficient, so that you would be having somewhere from the literature and getting it, and your V is the radial velocity. And the last one is your diffusion. So there are different means by which some of the particles gets diffused out. So there are when these forces act, so these particles get diffused out. So because of these forces, the sedimentation also occurs. So here the most important thing is the different forces are taking part in the process of sedimentation, or what are the different forces which takes part in the process of sedimentation. So there can be a question like mention the different forces, or take the different forces required for the sedimentation process. Okay, so then you have to write one is the centrifugal one, which is proportional to your omega square, then your R and the M. Then your buoyancy force, which is proportional to your omega square R and the M naught, that is your displaced mass. Then you have the frictional force, which is your minus AV, where you have the frictional coefficient and the radial velocity, and the force will also, sedimentation process will also depend on the diffusion. So these are the four important forces playing a role. And the sedimentation techniques are done in two ways. One is the velocity, and another one is the equilibrium sedimentation. Okay. The next one is speaking about the size of different protein molecules. So the diameter, so if you know that the diameter of a single hemoglobin molecule is around 5 nanometers, and the protein molecules can be very concentrated and at very highest physiological concentration of any protein are the crystallines in the eye lens cells, that can be greater than 50% protein by your weight. And then your fibrinogen is a large rod-shaped molecule that forms fibrin blood clot when activated. So these are just informations you can keep in mind. You may not expect any questions from here. So these are about mostly the size of the protein molecules. So the hemoglobin molecule has a size of around 5 nanometers. Your fibrinogen has a large rod-shaped structure and would definitely have a larger diameter, and it plays a role in the fibrin blood clot formation when activated. And your the protein molecules are quite concentrated. For mostly the highest physiological concentration of any protein are your crystallines in the island cells that has greater than 50% protein by weight. These are just informations you can keep in mind. Okay. Now coming to the next technique, which is via your recombinant means of purifying your proteins. So what we do in the first step is the DNA is first cloned, or we clone a particular gene of interest, and then we have our expression vector DNA, in which we, via some enzymes, that is the DNA ligase, we prepare a recombinant DNA plasmid, and this recombinant DNA plasmid is then transformed into the bacterial DNA, okay, into the bacterial cell. Okay. And then what happens is that the bacteria is able to produce the protein inside its say inside the cell of the bacteria. The protein is being produced. After that, the bacterial cell is disrupted, and then we get a protein. So the recombinant means is that what type of DNA can be cloned? You can make a DNA sequence, and then having a particular some cut sites and all, and then you also use those particular restriction enzymes to cut your or digest your expression vector. You also digest your DNA, and then you join the two. So first what you do, first you do a PCR, okay, with this, and then you clone the DNA. Then you actually digest your DNA, the PCR, the DNA, and then you also digest at the same time your vector DNA, and then you join the two. Say you join the two using DNA ligase, and then you get your plasmid in hand. Then you take some kind of competent cells, which can be your DH5 alpha competent cells, which are actually useful, which are your cloning vectors, okay, are your yes, your cloning vectors. And you have some expression vectors as well, like your BL21 and all. So these are the expression competences and the cloning competences from where you can extract your plasmids and all. So the expression vector, some of them are your PET28A or PET3A. Then you have the PET20, the different strains of PET and all. And some of the cloning vectors includes your PUF vector and all. Okay, so these are different information. So we do not require these at now. So what we now require now is the DNA, which has been cloned by us, and the expression vector. So we join these two via the DNA ligase, and we get the recombinant DNA plasmid. So we undergo the transformation process, after which the bacteria takes up the recombinant plasmid inside its cell, and then we plate them over the LB medium and the agar containing agar. So we plate them over some kind of a gel, that is the agarose, not the agarose, the agar plates containing. Also we when we do the plating, or when we give it some kind of growth, so these vectors contain some kinds of, you say, antibiotic resistant genes, okay. So only those bacteria which take up our plasmids, okay, or the plasmids of our interest, will be only able to grow, because the other cells will die, because of we are providing such kind of antibiotics inside our cells, okay. So only those from there we take up a cell and give it a growth, and then we can after we give it a growth, then we transfer it or we transfer it into a bigger condition, or where we have greater LB, greater nutrients, and then we induce that culture with IPTG, such that we can get our proteins. So the host cell has our recombinant plasmid containing our gene of interest, and under certain growth conditions, the protein is expressed, and then we harvest our cells. So here we can see some of the expression vectors. The prokaryotic expression vectors includes your pdest, then your pet, which is in your bacteria, and some of the eukaryotic expression vector includes like in your yeast you have the pic. Then in virus you have the baculovirus. In the mammalian cells we have the ped vectors. Okay. And then the other host systems, like what are the host systems? So that's bacteria or the prokaryote of the eukaryote, which is going to take our plasmid of interest or containing our gene of interest. Okay, so those includes your bacteria. If it's a bacteria, it is mostly your E. coli, or if it's among the eukaryotes, then in yeast we have the S. cerevisiae. In virus we have some insect cells. In the mammalians we have the HEC 293, that is the human embryonic kidney cells. Okay. Okay. Now what we do now, just as I was speaking. So what we have done till now, we have the plasmid in our hand containing we have the recombinant plasmid or the recombinant cell. We have this one, the DNA plasmid, the recombinant DNA plasmid. Now we undergo the transformation, and then the next day we get colonies. So from the colonies we give it a overnight culture. So this is the overnight transformed bacterial culture. That is when we give it a growth from the colony. So what we do, where do we give it a growth? So what is this culture about? This is the your LB or the Luria Bertini broth. So or the growth medium. So what does this growth medium actually contain or actually influence these bacterial growth? These growth mediums actually influence this bacterial growth. So these have a particular pH. Then your this requires a particular temperature for your growth as well. So some needs an incubation period of 37. Some request even lower like 30 or 24. Some requires even less like 15 or 12 degrees, or sometimes we give it at very high temperatures like 40, 44. So these are the different temperature factors which also is required by the different kind of cells or the different kinds of bacteria for the expression. Okay, so also these growth media has particular nutrients which are required. So mostly these nutrients are you can say it contains yeast extracts. Then you have some salts like your NaCl, then you have tryptone and some kinds of amino acids and all, which are actually not produced by the bacteria, and the bacteria requires these for the growth. Okay, so we give it an overnight culture, and after what we do the next day we give, because this is called as the primary culture. So first we give the primary culture from which is given for overnight. The next day we we transfer these culture into a greater volume. We increase the volume of the growth media, and we transfer this primary culture into that huge volume, and we call this as the secondary culture or the 1% culture. And then we incubate it at say I'm saying at 37 degrees centigrade. And then after this inoculation process is done, the cells grow because it has got more and more of culture, more and more of nutrients, and it's hungry for more food, and it gets it and it grows more and more. So then we check the OD. So the optimum time when we should actually induce this IPTG is at when we get an OD 600 at around 0.6 to 0.8. For 0.4 to 0.8 is okay, but 0.6 to 0.8 is much more optimized and optimum OD for actually your inducing of the IPTG. So we check it, we take the 1 ml of the culture, we do the blank first with the normal LB or the normal growth media, and then we take 1 ml from the secondary culture and check the OD in the some kind of nanodrops and all. And then when we see that we get a optimum OD say 0.6 or 0.7 or 0.8 or something, then we induce our IPTG. Then the IPTG induction is done, and after this IPTG induction is done, the protein expression starts. So before this IPTG, the cell is growing, and only after the IPTG induction is done, it um, the protein starts expressing inside the cells. So what is this IPTG actually doing? So it is actually mimicking the allolactose, that is the lactose metabolite, and but it does not it is not a part of the metabolic pathway. It what it does, it just triggers the transcription process of the lac operon, and it is used for the induction of the protein expression. So it is so when the repressor binds to the your say the lac operon, the promoter region, then the protein cannot express. So when these IPTG is being induced, what it grabs the repressor, allowing the operon to actually function and express our protein. So this is the function which is being played by the IPTG. So IPTG is isopropyl beta D1 thiogalactopyranoside. And then after the induction process is done, our protein starts expressing, and then we incubate it at, say, whether we want to incubate it for overnight or say how long, that depends on what type of proteins and what is the half-life of the protein and all. So when we incubate it at, say, 37 degrees centigrade, because we have to keep the conditions also same, so we give it incubation, and after that we actually harvest the cell. What is harvesting of the cells? We centrifuge the entire culture, and we remove the supernatant from that. We take the cells, and then when so here the pellet is actually important, not the supernatant, because we are getting the cells containing our proteins which has been expressed inside the bacterial cells or the eukaryotic cells and all. And then we harvest the cells, after which we perform the which we get our protein after we lyse the cells. Okay, so first the primary culture is given, then the secondary culture is given. After that, we when the OD reaches at somewhat 0.6 to 0.8, then we induce it with IPTG. And after this induction process, the protein starts expressing inside the bacterial cells. After a certain amount of time, we centrifuge it and we collect the cells and store it. This is called harvesting of the cells. So we need to keep this thing in mind, that only after the addition of IPTG our protein expression will start, and we can keep these things in mind as to what is the role which is being played by IPTG. So it is a molecular mimic of the allolactose, and it triggers the transcription of the lac operon. Okay, so I will be speaking about purification of proteins. Let's take a 5 minutes break. After that we can start. Okay, so let us start with the purification of proteins. So, in the purification of proteins, so how can proteins be purified? So, one is via your differential centrifugation, then you have the differential salt precipitation, then we have via your differential solvent precipitation, then we can do it via preparative electrophoresis and also by some column chromatography. So what does this differential centrifugation speak of? So like before we were doing with the different cells and all, so this is the entire figure which is given. So I've spoken a lot about this centrifugation techniques. You can go through the this picture, and the same thing is written as to how the speed will actually lead to what kind of pelleting. So here separation of proteins are done on the basis of size, mass and density. So it is the function of the size of the protein and the speed of the centrifugation. So it speaks on the function of the size. So it is also the same, the size of the protein which you are using and what is the speed of the centrifugation which you are using. And so what is one of the disadvantage is that it gives us a rough separation, and it is basically that we are fractioning it. We are just fractioning it. Okay. So the next process is dialysis. So dialysis is when we do some kind of buffer exchange or when we are dialyzing it. So we have a dialysis membrane, then we have a solvent with which we want to exchange our say or change our buffer with. So this solvent can be just mere water as well if we want to just remove the entire of the salts and all. And the inside solution is mostly concentrated. The vice versa process is sometimes also true if we have to increase the salt concentration inside our protein and all. So here we have the protein. So inside this we have the protein, and then this is some kind of buffer provide maybe with less ionic strength and all. So that is different altogether, but then this is if this is a concentrated, the inside one is a concentrated solution and the outside one is a less concentrated one. So the principle here is based on our osmotic pressure, and it allows us to get rid, and at the same time it allows us to get most of the salt ions as well if the inside one is a the, what you call the less concentrated one or the dilute one. Okay, so it also allows us to get rid of the most salt ions, uh, but the entire of the salts cannot be removed. Can you tell me one reason why the entire of the salts cannot be removed? So first is the it undergoes some kind of osmosis and then the salts are out. But why it is not possible to remove the entire of the salts? Can one of you tell me what can be the probable reason for it? Why are we saying this? That the entire salts cannot be removed. Any one of you can tell me. Just think why the entire salts cannot be removed. No one? Okay, so if you see that when the salts are going out, the outside of the solution is also getting concentrated, or also um, the solvent is also getting increased ionic strength, or the ionic strength of the solvent is also increasing, and there would be one point in which there would be an equilibrium condition, because the ionic strength the salts are going out, and then because there is an equilibrium in between them, till that moment the salts would be going out when the inside concentration would be higher or lower than that of the outside concentration. But if they two become same or equi-concentrated or having equimolar concentration, then there would be no more exchange of salts or exchange of ions between these two conditions. Okay, so therefore it is not possible to completely remove the salts. Okay, so that is why just before the equilibrium condition is reached, the salt exchange or there is exchange of ions. But when the equilibrium condition gets reached, then no more salt exchange or ion exchange occurs. Therefore, it is not always possible to remove the entire of the salts or the ions. The next one, I have spoken about it in the previous class as well, about column chromatography. So the most common and the best approach to purify large amounts of proteins is via this type of chromatography, and it also helps us to achieve highest level of purity in largest amount as well, in with low effort as well, and it has the lowest possibility of damaging our protein, and it is the mostly used method for different in different pharmaceutical industries as well. So it has different advantages. It is the best approach for purifying proteins as it achieves highest level of purity with the lowest effort, giving less damage to our proteins, and mostly used in the pharmaceutical industries. Okay. So the three different types of column chromatography I have spoken before is a gel filtration chromatography, then we have the ion exchange chromatography and the affinity chromatography. Okay, so the gel filtration chromatography, it is separated according to the differences in their size as they pass through the hydrophilic polymer. So the polymer beads are mostly composed of cross-linked dextran, or we call as the dextrose, right? So the dextrose, which is highly and uniformly porous like the Swiss cheese, like this cheese, and this is uniformly and highly porous, and the larger proteins comes out fast because it can't fit in the pores, and the small proteins comes out in the end because they get stuck in all the pores. So this is the general principle behind the gel filtration chromatography. So what happens over here is that this is our porous polymer beads, and the protein mixture is added to the column, and then it passes. Okay, and then the protein molecules. So then the it gets stuck in the different kinds of pores depending on the size and all, and the protein molecules separate out by size. So the larger molecules pass more freely, appearing in the early fractions, and the smaller fractions will get eluted later. The ion exchange chromatography is on the basis of charge adsorption. It has highest resolving power, highest loading capacity, widespread applicability, and it is the most frequently chromatographic technique which is used for protein purification. So this is a kind of a cation exchange chromatography where we have a lot of negative charges and which gets attached to. So these are the beads which are DAE cellulose, made of DAE cellulose, and then so what is a cation exchange chromatography? I've spoken. You people only can tell me what is a cation exchange chromatography. So what can be separated in a cation exchange chromatography? Now this type of answers you people only can give me, because I've taught in the last class, and in the few days you would be having exams, so I guess you people are revising as well. the previous lectures. So it's not always possible for me to tell everything because it's just a 2 hour class, so you have to go through the lectures as well, mostly. Tell me what happens in a cation exchange chromatography. One of you can tell me what happens in a cation exchange chromatography. So what are the beads made up of in a cation exchange chromatography? Anyways, so in a cation exchange chromatography or in an ion exchange chromatography, what happens is that the beads are made up of the other charged ions. So if the beads are made up of, or the resin which we are using is made up of a negatively charged ion, then it will actually grab the positively charged protein molecules. Okay, so if the protein has a positive charge, it will get attracted to the negative charge. So but what happens when the protein is negatively charged? It comes out faster. So in the cation exchange chromatography, you have a negatively charged resin and then your protein is positively charged, and then you pass through the protein, and then the first your so this is an example of so this is an example of your anion exchange chromatography, because you have positively charged resins and then the negatively charged proteins gets attached to it. So firstly you get the positively charged ones, but the negatively charged ones gets eluted later. So this is an example of the anion exchange chromatography, but this one is probably, this is not an example, this is probably the principle. As the protein mixture is added to the column containing our cation exchanges, and then what happens is that the oppositely charged proteins gets attached to it or attracted to it, is absorbed by it, and then when you elute it, but the same charged proteins would be eluting faster. So if it's a, so another time I'm repeating, if it's the cation exchange chromatography, the resin is made up of negatively charges and your protein is positively charged. So if you have negatively charged proteins, then they will elute faster because they won't get absorbed or attracted to it. But if it's an anion exchange chromatography, the resin is made up of cations or positively charges, and your protein, only your proteins which are negatively charged gets attached to it, and positively charged ones gets eluted faster, the negatively charged ones gets eluted later. So these things you have to keep in mind, because there might be a few questions regarding this, but I expect you people to at least give me a few answers as well, because this is a live session and you people also have to interact with me. Anyways, but you keep these things in mind, that would be enough for your exams and the assignments. So coming to the next type of chromatography, which is your affinity chromatography. So this is an adsorptive separation in which the molecules to be purified specifically, and it reversibly binds or gets adsorbed to the complementary binding substance, which is a ligand which is immobilized on an insoluble support, which we call as the resin. So in this case, the purification is 1000 times better than a single step, and this is mostly the preferred method if possible. So in the previous class I have spoken about the conditions which the ligands needs to have, or the characteristic features of the ligands and what type of resin you can use and all. So you can go through that lecture as well. In short, this is an affinity chromatography where it has a particular affinity for a particular ligand and all, and this should not have very tight binding because you need to elute it at the end of the day. So it should be sufficient such that it can bind, and at the same time it can get eluted at the same time. So the protein mixture is added containing a polymer bound ligand specific for the protein of our interest, and then the unwanted proteins are washed away through the column, and the protein is eluted using some kind of solution or a buffer which will help in the elution of our protein. So we need to keep in mind that the protein is not covalently bound to the ligand. So it has a good binding but not a very tight binding. Okay. So coming to the next type of thing which we can do is the isoelectric focusing. So here we separate the proteins based on their pI. So when our pH is so we have a particular buffer solution. If the pI of the buffer is less than that of the pH which we are using, then, or if the pH of the buffer is higher than the pI, then the protein would be negatively charged and would be moving towards the anode, whereas if the pI is greater than that of the pH, then it would be positively charged and would be moving towards the cathode, or the negatively charged side. So this is isoelectric focusing. The next one is the polyacrylamide gel electrophoresis, where we separate the proteins based on this size using the electrical current. So having different sizes and molecular weight, we separate them. So these are the largest ones and these are the smallest ones, because the largest ones would be passing very slowly, whereas the smallest ones would be passing quickly or flowing quickly. So this is a vertical way of actually separating out our proteins. And first what we do is we load the protein samples in the gel. So these gel can be of two types. One is the SDS page and the other one is the native page. So in the native page we actually separate the proteins based on its PI, as well as whether the protein exists in what type of native form does the protein exist in. We can check that. But when we perform an SDSP, the protein acquires a uniform negative charge, because of which we are separating it based on their size and the molecular weight. Okay. Now coming to the next type, or a new type of electrophoresis, that is the 2D page or the preparative electrophoresis. What we do in the first step, we separate the proteins based on isoelectric focusing, that is based on their PI. Whereas on the other hand, we perform the SDS page. So in the first way, we separate them isoelectric focusing, and in the this one is your isoelectric focusing, and then in the down way is the SDS page. Okay, so we get this type of image. So from this we can also comment two things: one on the native structure of the protein, the size of the protein, as well as on the pH or the PI of the protein. So from these two informations can be gathered from the preparative electrophoresis. Okay, so I think that's all with the theory part. Now we can solve a few questions. Okay, so yes, you people can answer now. So what does the first question say? Which of the following factors influence bacterial growth in the growth media? Which of the following factors influence bacterial growth in the growth media? Yes, so D is the right answer. All of these are important. Just as I was speaking, the temperature also plays a role. There needs to be sufficient amount of nutrients, as well as the pH of the growth media is important. So option D is correct, 1D. Question number 2 says the lysogeny broth does not contain what? What does the lysogeny broth does not contain? So I was telling it needs to have yeast extract, also some salts and metal ions have to be present. You need NaCl as well. Also tryptone has to be present. What you do not require is agar. So when do you require agar? When you are actually forming plates on which you have to actually culture your bacteria, or you need those colonies after your transformation process. Okay, so then you actually need those agar plates. So but for the lysogenic broth we do not require what is agar. So for question number 2 the right answer is D. The agar is not actually important in the lysogenic broth. Okay, which of the following elements is the micronutrient for bacterial growth? So if you actually see what are the elements required for the bacterial growth, it is actually your carbon, then you require your nitrogen, because if you see that these kind of growth medias have your amino acids, and these amino acids will actually contain more of your carbon and your nitrogen, because you know the structure of amino acids. So carbon and nitrogen has to be present. So these would be basically your macronutrients which are required for the bacterial growth. What is a micronutrient over here that you also require? So basically it is cobalt. Okay, so question number 3, it is C. Cobalt is a micronutrient which is required for the bacterial growth. Okay. In order to lyse cells, sonication is used. The process breaks apart the cells by. So what type of energy do this sonication process use to lyse the cells? No? This is an easy answer. What does sonication, what type of energy is used in sonication? Yes, right. Yeah, that's correct answer. So question number 4, it's C. It uses sound energy to actually lyse the cells. Question number 5 says a protein will be positively charged when pH less than Pi, pH greater than Pi, pH equals to Pi, does not depend on Pi. B. Okay. So when your pH is greater than Pi. So what happens when your pH is greater than Pi? Say your Pi is 4 and you use a pH 6. So if your pH is greater than Pi, your proteins would be negatively charged because your Pi is greater less than that of pH. So the Pi has to reach for the pH, and when the Pi has to reach for the pH, then your proteins gets negatively charged, because when you're increasing the pH more than your Pi, then your hydrogens gets abstracted and you get negatively charged over the protein. So pH greater than Pi means your proteins would be negatively charged. When your pH is less than Pi, only then the Pi has to, means the protein has a Pi say 6. You're using a buffer of 4. Then your Pi has to go down and have to collect a few hydrogens and go down to lower the pH. Okay, so then your protein would be actually positively charged. You got it? So when pH less than Pi, so in order to lower, to reach that pH, it has to collect a few hydrogen ions or the hydronium ions. Only then that pH can be reached. So collecting those hydronium ions means actually giving the protein positive charge. So the pH has to be less than that of the Pi for the protein to be positively charged. Okay, what happens when pH equals to Pi? What happens to the protein? Can you tell me what happens to the protein when your pH equals to Pi? Because there can be a question, what happens when the pH equals to Pi? Yes, that is correct. Very good. Okay. What does question number 6 say? In size exclusion chromatography, proteins are separated based upon their. What do you separate, or how do you separate in size exclusion chromatography? Size and shape. That was quick and correct. Okay. So question number 6, we separate the proteins based on their size and shape in size exclusion chromatography. Okay. Good. Question number 7. A mixture of three proteins of sizes 50 kilodaltons, 30 kilodaltons and 12 kilodaltons are separated by size exclusion chromatography. What is the order in which they will be eluted out? 50 kDa followed by 30 and finally 12. All of them comes out together. 12 followed by 30, finally 50, or 30 followed by 12 and finally 50. Slide is not visible. Uh, is it visible now? Okay, good. So in which manner would these proteins be eluted out? Or how does proteins elute out in size exclusion chromatography, right? So first the higher molecular weight protein would be eluted out, after that the lower one, and then the lowest one. Yes, so 50 kDa followed by 30 kDa, and last you would be getting the 12 kDa. Okay. Question number 8. Imidazole can bind to which of the ion? Ion or ions? Or when do we use imidazole? What do we use imidazole for? Right. So it is nickel, because we use imidazole when we are performing nickel NTA chromatography. Okay, nickel NT affinity chromatography. So imidazole would be binding to your nickel, and then your protein would be eluting out. So A is the right option. Good. Question number 9. You have expressed proteins in bacteria and lyse the cells. Okay. So we have expressed proteins in bacteria and lyse the cells. How will you check if your protein of interest is present in the cell lysate? So what do you do? First you express the protein in the bacteria, and then you harvest the cells and then lyse the cells. So how would you check that the protein is in your cell lysate? What kind of experiment would you do? What is the simplest way you can actually check your protein is there in the lysate or not? You said D, analyze the lysate by circular dichroism. It actually speaks on the structure of the protein, you know. So if you do circular dichroism, so CD spectroscopy, it will actually speak on the different secondary structures which are present in the protein. Okay, but how will you check whether a protein is there or not? When you get your protein, you do circular dichroism, that is okay. Then you can tell that these are the number of alpha helix or the beta sheets present. So the simplest way you can do is just run it on a page. You do SDS page, you run it on a page, and then you check, you run the gel and then you stain it, then destain it, and then you check whether your protein is there or not. That's the simplest and the easiest way you can do it. So just take the lysate, add a dye and SDS to it, heat it, run it on a gel. Simple. So for question number 9, it's B. Okay. Question number 10 says, your protein of interest is 90 amino acids long. What is the approximate molecular weight? So first tell me, what is the average size of an amino acid? The average weight of an amino acid. First tell me that. Then you can calculate it easily. Yes, very good. So it's 110 Daltons. Now the answer is easy. What is it? See, 9.9 kilodaltons. Aarti, do you agree with him? Or do you have any other answer? Definitely you should not be having. This is an easy answer. Okay, good. So 10C is the right answer. Okay. Question number 11 says, protein absorb at 280 nanometer due to the presence of which amino acid among the following? Right, so both B and C are correct. Actually, your tryptophan, tyrosine and phenylalanine, all of these three amino acids are responsible for the getting a peak of your protein at an absorbance at 280 nanometer. So why did you answer only C? It's B and C both. Okay, you have to keep these things in mind. Sometimes you may not be said that it is an MSQ. Probably you would be given that this is an MSQ, so you have to go for both. But yes, it's okay. If it is an MCQ also, I guess B and C would be right answer. Okay, so okay. And anyways, it's B and C, tyrosine and tryptophan. Okay, question number 12 says, 280 nanometer, a protein solution has an absorbance at 280 equals to 9. And the molar extinction coefficient of the protein is 11460 molar inverse centimeter inverse. What is the concentration of the protein? Tell me the answer. After that, I would be doing the calculation. So what was the formula? A equals to epsilon Cl. Right? Oh, you did the calculation. Oh, no, I need to do the calculation. Okay. Okay, let's wait if we get any other answer. But remember one thing, always check with the units, because that can actually be very tricky. So you do quick calculation, that is okay, but sometimes you might lead to a wrong answer. So always check with the calculations, because I guess the answer which you're telling is probably in molar, but this is in millimolars. Although I do not know, I haven't done the calculation, I need to do it. So what is it saying? So it's 9 is equal to 11460, and you know that the length is 1 centimeter, so I'm not even considering that. So then you have your C, and then your C is equal to your C is equal to your 9 divided by 11460, that is in molar. Okay, so what is it going on? What are you getting? You're getting 0. I think 000 yes, 79 molars, but the value is given in millimolar. So what is it? How much? 0.79 millimolars. Okay, So do the calculations. You people know the right answer or how to do the how to solve the sums, that is right. But don't just jump because that's an easy question, because these units would be tricky. I'm telling you people, these units would actually play around with you. So be very concentrative when you are solving these easy sums, because these are very easy sums and very scoring. You do not have to keep anything in mind, just you have to go there and use the calculator if you are allowed to use it. I do not know though, but if you're allowed, this is just simple calculation, but these units would be very critical, so do very cautiously. Okay, so for question number 12 it is B, so 0.79 millimolar is the right answer. Okay, question number 13, for the salting out of proteins, which of the following salts is commonly used? Okay. Oh, that was quick. Okay, let's wait for Amardeep to answer. Okay, so both of you have said C. I also say C is the right answer. So ammonium sulfate would be used for the salting out of proteins. Okay, question number 14. To determine the size of a protein, which of the following techniques can be used? Which of the following techniques can be used to determine the size of the protein? Size exclusion or SDS or analytical centrifuge or any of the above? Both of you said D. Yes, any of the above techniques can be used for determining the size of the proteins. You can check back with my slides, and how you can determine them, that also you can understand. So D is the right option. Okay. Now coming to question number 15. So this is the sequence of a protein which is given to you. Which of the following statement of the above protein is true? So mostly I won't be pressurizing you with this question. I am telling you the answer of this. First, what you need to do, if this type of questions I am sure won't be there. So because this is actually not okay to actually, you would be counting the amino acids and then calculating the molecular weight. Probably you would be able to calculate the molecular weight. But how would you be commenting on the pi of the protein or the extinction coefficient of the protein? Okay, or whether how would you understand if such a protein would be soluble in a buffer of pH 10? uh, no, in the exam this type of question is not possible. I have just kept it because this was a previous year question which was given, so I have just kept this question. But this type of questions you will definitely not be getting in your exam. And if there is a question, and this would mostly be an MCQ question where you would be have to count the amino acids. I don't you do not expect such a question. You would be given the length of the amino acid and then you have to calculate. But as of now, what you have to do in the future, if you get this type of questions, not in the exam, anytime, what you have to do, I'm telling you that. So you have to copy such a sequence of the protein sequence and then paste it in a expressive protparam input box. So there is a tool called the protparam tool under your expressy channel, and then you have to paste this sequence in the toolbox, and then you have to click on the compute parameters. So I don't know if I can show you this. Anyways, so you have to copy this entire sequence and then paste it, and then you will be getting the parameters and all of these, where you would be getting the pi of such a protein, you would be getting the molar extinction coefficient of that sequence, and then also uh, you would be getting the molecular weight of the protein, then number of alanines are there, the number of amino acids, each amino acid present over there, etc, etc. So just because it was an MSQ question in the previous year, so The molecular weight of the protein is 10.5 kilodaltons. If you calculate it, you would get it. The pi of the protein is 10 and the molar extinction coefficient is 22.46 millimolar inverse centimeter inverse. So if you paste it in Expressy or in the Protparam tool, you would be seeing that A, B and C all three are correct. But in the exam, this type of questions won't be coming for sure. Okay. Coming to the next question, question number 16. The protein in the previous question is in a buffer of pH 8. It can be purified by. So what information you can gather from the sorry. Am I audible to you people? Because I am having a network issue. Can you just verify if I am audible to you people? Hello, am I audible? Okay, good. Right. So in the from the previous question, you can see that the pi of the protein is 10. So the protein in the previous question is in a buffer of pH 8. It can be purified by. So you know that the pi of the protein is 10. So what it can be purified by what type of chromatography? What type of chromatography can be used for purifying such a protein sequence? So you can see that here the pH is actually less than your pi. So what would be the charge on your protein? Okay. B. Any other answer? How many people? Yes, we have another one. What do you say, Amardeep? What would be the answer to this? Aarti, any other chromatography can be used? Do you think so? Okay. You say A and B. You are saying D. Okay. See, first thing is that the pH is less than pi. Okay, so the proteins are positively charged. Hence, you would be definitely using a cation exchange chromatography. Okay, the other type of chromatography which you can use is the size exclusion chromatography, because this can be size exclusion chromatography can be used for actually purifying these proteins. There may not be a competition between proteins, but you can use size exclusion chromatography for separating out your proteins. You cannot use an anion exchange chromatography because it's positively charged. And the other thing is that there is no histidine tag. If you check that, you do not have any histidine tag over here. Either it is present in the N terminal end or the C terminal end, but there is no histidine tag. So you cannot use means you cannot use a nickel NTA column for actually performing affinity chromatography. So A and B are the right options. That is a size exclusion chromatography and the cation exchange chromatography. Okay, the next one is, which of the following techniques is best suited for estimating the overall type of secondary structure of a protein? I was speaking about it a few minutes back only. Tell me, which of the following techniques can be used? So for estimating the overall type of secondary structures, yes, we use circular dichroism for checking how many alpha helices or beta sheets are present in that. Yes. Okay. The last set of questions, we have three last questions. The three-dimensional structure of a protein can be determined by. So circular dichroism uses determines your 2D structures. Size exclusion chromatography purifies your protein. So the three-dimensional structure of a protein can be determined by A. Yes, you can use NMR spectroscopy and X-ray crystallography. Correct. So very good. So you can use both NMR spectroscopy and X-ray crystallography for determining the 3D structure of a protein. Okay. Question number 19. Particle size and sample homogeneity can be determined by. No? So okay, so you can actually determine the particle size and the sample homogeneity via three techniques over here. Yes, one is definitely your analytical ultra by using an analytical ultracentrifuge. Also, you can do it if you perform dynamic light scattering and also size exclusion chromatography. So your A, C, D, all three are correct. You can do it using dynamic light scattering, analytical ultracentrifuge, as well as your size exclusion chromatography. So your A, C, and D, all three would be correct answers. Okay, coming to the last question. A protein without any affinity tag is being purified from sonicated crude lysate. Which of the following is the best purification sequence? So a protein does not contain any of a affinity tag, and it has to be purified from the sonicated crude lysate. So what would the purification sequence be such that you get the best of your pure protein? Okay, so Arti, you said D. Let's wait for Amardeep. What do you say, Amardeep? Okay. So if we see options A and C are wrong because your protein of interest does not have any histidine tag because it is without any affinity tag. So you cannot use the Ni2 plus column. Okay, so these two are out. Option B is also wrong because the sequence or the order of purification is wrong. First you do ACC, then ion exchange, then salting out does not make a sense because salting out at the end will actually make no sense. So first you need to do salting out, then you need to do ion exchange and ACC. So what you do, the crude lysate which you obtain after sonication, you subject it to the salting out, and what will happen, the proteins which precipitate out are removed and then separated from the proteins which remains in the solution. Now these what you get in the supernatant is then you perform ion exchange chromatography on it, from which you can separate some contaminant proteins from the your protein of interest, and then finally in the final step you can purify your protein of interest via the size exclusion chromatography, and then you would be getting your pure protein. Okay, so That's all. I'm done from my part. Thank you. If you people do not have questions, we can end it over here.